Prebiotics, basically food for the good bacteria, as well as combined pathogenic organisms, some of those. Probiotics are the good bacteria. And these are names that we hear like lactobacillus and bacillus.
Good morning, everyone. Welcome to the Poultry Podcast Show. I'm your host, Karen Grogan, and joining me today is Dr. Theresia Lavergne with Natural Biotics. Thresia is a nutritionist by trade and spent many years on the faculty at Louisiana State University and her work there in extension. So maybe we'll touch on that. But in her current role, she helps support a number of products that help support the microbiome of the chicken. So we're going to focus on that as our topic today. So welcome to the show, Thresia.
Well, thank you very much. Good morning. Whatever time, I guess it's morning when we're recording.
It is still morning when we're recording, but people will listen to us probably all hours. I've talked to several colleagues and they're like, oh, I listen to you when I'm driving. You know, they're driving from wherever they're going for the day for whatever. So I think a lot of people listen to us in the car. So no telling what time.
Thanks to everybody who's going to listen or who will listen no matter what time it is.
Excellent. So let's start a little bit. Thresia, why don't you kind of give us your background and how you got to the job that you're currently in?
So you already mentioned I'm a nutritionist. I have a bachelor's degree in animal and dairy sciences. And then for both my master's and PhD is when I focused in nutrition for the monogastric, actually both swine and poultry. And I went to school at Auburn, Mississippi State, and LSU in that order.
You have officially toured the SEC schools and worked your way west.
Right. And I actually live a little further west now. I actually live in Texas now.
Oh, just keep moving west.
But still south.
Excellent.
So, yes, most of my career, I was at LSU in the School of Animal Sciences as a poultry extension specialist, as well as, professor, where my extension work, of course, working with the industry, you focus on what your industry needs and where you can help them the most. So I actually did a lot of litter management, mortality management, type of work, as well as biosecurity training, just kind of overall, as well as I worked with the 4-H and FFA programs. So I worked with all ages, all kinds of people, as well as, being in the department where I was able to do some research. I had obviously mostly an extension appointment, so it was more field type trials. But towards my last years there at LSU, I was able to get back into some nutrition research and have some graduate students. And then several years ago, I decided to go to industry. And so since I've been in industry, I've been supporting probiotic and prebiotic products as a technical service manager. And then about a year and a half ago, I went to Natural Biotics, where I am now. Again, still supporting the same types of biotics, prebiotics, probiotics, right, and now postbiotics. So in addition to technical services, I do coordinate our poultry research. So this has been great because now I've gotten back deeper into research and development, as well as, working out with the industry and technical services.
Natural Biologics is using cutting-edge science to dig deeper into the poultry health challenges you face. By gathering scientific evidence, they identify the most effective combinations of natural ingredients that improve animal health. Visit naturalbiologics.com/poultry to see the newest research in both turkeys and chickens.
So I think in your description there, you covered a great spot for us to start our conversation in terms of all these biotics, in terms of prebiotics, probiotics, and then postbiotics. So maybe that's a good place to start if you could explain sort of what each of those targets and how they work differently in whatever gut of whatever your species, be it poultry or swine. So what's the difference in those and how do they work?
Okay. So prebiotics are generally the food source for the good bacteria. So prebiotics, a lot of the prebiotics we use in feed are coming from the yeast cell wall, like the mannan oligosaccharide, or MOS would be the short term for it. They are relatively indigestible to the host, the poultry, and stay in the gut and serve as an excellent feed source for the good bacteria within the gut. In addition, because they're made up of mannose, they are able to bind to mannose-dependent binding pathogens like salmonella and E. coli, and they can bind to these pathogens, prevent those pathogens from attaching to the gut lining, colonizing in the gut, and causing some issues. So prebiotics, basically food for the good bacteria, as well as combined pathogenic organisms, some of those. Probiotics are the good bacteria. And these are names that we hear like lactobacillus and bacillus. Primarily in our feed, we're using a type of bacillus strain. There are some others out there as well, I guess, but over the years, our primary one that we've been feeding as a probiotic is a bacillus. So we're putting a good bacteria in the gut to take up space in the gut, do all the good things, because those bacillus do produce products that can be that will be antimicrobial, help with they can produce some enzymes, help with some digestion of feed ingredients. So they're good bacteria that just contribute to the overall gut health of the poultry. And the other is postbiotics, which is a newer kind of class of feed additive or tool we have for gut health. And these postbiotics are compounds produced by mostly probiotic strains or yeast strains. They're end products. And can, produce, they can be have some enzymes in them, some amino acids, some nucleotides. So they produce products that are good for the digestion, overall gut health of the poultry. So prebiotics, probiotics, postbiotics.
Perfect. Okay. So we know, I think everybody throws those terms around a lot. And unless you work in that sector, or you're a nutritionist, or you're trying to figure out how to add these things into formulations, it's good to always clear that up when we're starting a conversation about that. So in terms of trying to sort of manage the poultry microbiome, it's pretty complex. So how do you get these to survive, like, let's say, feed manufacturing? That's one battle is, how are they formulated so that you can survive the feed processing? And then, how do we measure, the impact they're having? I think that's another thing that is a little difficult to quantify is, we want this impact and we want, an end result. So how do we evaluate those?
Okay, so I guess first to go back to how do they survive the process of what we do in poultry feed, pelleting, crumbling. First, the prebiotics, or, again, I go back to the yeast. By far, the majority of what we use are from Saccharomyces cerevisiae. And those products are not alive for most of them. There are some live yeast products, but for the most part, these prebiotics are not live. So we're not killing them as we would go through our normal heat process. They're very, very heat stable. So they make it through the whole process that we use in pelleting and crumbling chicken feed. Probiotics now, we do have to be a lot more careful there because they do not withstand a lot of the heat or the levels of heat we might use in our feed manufacturing. But like a bacillus is a spore former. So it's the spore form that survives the heat and then is activated again inside the poultry. Postbiotics, again, are not live. They are compounds that were produced by these yeast or probiotic strains. So again, they're not live and they're good to make it through that heat process as well. Now, as far as evaluating their effectiveness, Obviously, there's going to be many ways we can do that. And a lot of people have their own different ways of looking at it. Being a nutritionist and one who's done a lot of kind of feed 'em, weigh 'em type research, really kind of, I like practicality and how it can all fit together. So, that's the first thing we start with. We do a growth study. We look at their average daily gain, obviously feed intake, because we need to make sure they're eating. And then, of course, we do use those metrics to calculate feed conversion. And we typically do see an improvement in feed conversion by using a probiotic, prebiotic, especially because they're improving the overall gut health. And, just maybe reducing the pathogen load like they're supposed to do as well. So an overall improvement in health, because health is definitely, and especially overall health and especially gut health, are tied to performance. We have a dysbiosis situation, an imbalance in our microbiota, shifting towards more pathogenic bacteria. Obviously, our growth performance and health is not going to be as well or as good as it can be. So that's one measure we have to look at the effectiveness of these products or biotics, is just growth trials. Where we're getting today, we can also look at specific pathogens. Whether we're having to sacrifice animals at the end of research trial or in live production at certain stages of growth, we're able to take that digestive tract and look at the pathogen loads and overall microbiota within there to determine, obviously, our load of pathogens, what pathogens are in there, as well as, on the other hand, what good bacteria or bacterial biomarkers are present. So we can again, and again, I'm going to go back to the prebiotics just as an example. If we're looking for, are they controlling salmonella and reducing the colonization, right? We can collect ceca and analyze those for salmonella prevalence or load, and then, or we can use cloacal swabs and do the same thing, analyzing for salmonella load and prevalence. And so we can actually take measurements to see, are they reducing the pathogens? And then along with, are we looking, our growth performance? Are we getting optimum growth performance through these?
So in terms of things like that, like the pathogen control, are they actually like a challenge trial? Like, so you have birds that are fed, whatever, the prebiotic, probiotic combo, or some, a product, they actually get challenged with a known, say, salmonella strain. And then you look at, the difference between two treatment groups. Are we talking that kind of a research, or, more kind of like field trials that we're looking at just naturally occurring salmonellas and looking at the difference between, or maybe all of the above?
It is all of the above. And I've been involved with all of the above, right? More obviously in a research setting, we can do those challenges with salmonella and look at feeding, obviously you keep a control to complete your experimental design and have something to compare to with these products you're adding into the feed. And yeah, you can do the challenge and look at the salmonella loads over a period of time, or, whatever your point is that's most valuable for whatever data you're collecting. But out in the field, we'll just, or just even some research trials, just kind of I guess we call it a natural challenge, using built up litter. That can be your challenge within a research situation, but definitely in the industry, we do that. So there's a lot of salmonella in the environment to begin with, whether it comes from the litter or, unfortunately, rodents, pests that come into the environment of the poultry can bring the salmonella. So more out in the field trial work that I've done, it's been, implementing a product, prebiotic, probiotic, into the diet, and looking at different periods of time, the salmonella load, if that's what we're evaluating, prior to going on product, and then different periods after. So like I said, either way, you can obviously in research, we challenge, we're looking for modes of action, definitely demonstrating that reduction in effectiveness of the product. And then we go out into the field, whether it's just a field trial or actually working with a customer that's trying to evaluate the effectiveness of these types of products that they're using.
And in terms of that, like all the way through to processing, like you're, evaluating like carcass rinses, like you're looking at stuff that goes all the way to an endpoint, or more kind of using this as like a pre-harvest control point?
Right. Yes, excellent question, because we're always asked in live production how this is going to affect processing, what's going to happen. So these products are in the feed, obviously. So definitely we're looking at live production and pre-harvest, right? Getting that bird, and a broiler, turkey, whatever we're growing, getting that bird to its endpoint to go into processing as the lowest, with the lowest salmonella. It could, we're trying to lower that. Now, once that bird leaves the farm, everything changes, right? They're hauled. Even with feed withdrawal on the farm, that's a stressful time. And so some of these birds they'll have an increase in salmonella at that time as well. But then, they feed withdrawal, they go transportation to the processing facility, all the factors within that processing facility. So these are really just a part or a piece of an overall salmonella control program. And again, if we're talking about the prebiotics or the yeast, type ingredients, it's just a part of it. But we want to do our part in pre-harvest to make sure that bird has the lowest amount of salmonella, or salmonella free would be great. But we try to do that pre-harvest and make our contribution, because it is an overall, production scheme from live to processing.
I think that, so in terms of like trying to evaluate the microbiome and everything, the tools we have in terms of like, like you said, like detecting their metabolites, or, all these like proteomics and genomics and all of these omics, in terms of evaluating what is actually there and what's doing what, has just evolved immensely. Even the past like 5 years, like the speed at which we can do it, the preciseness at which we can do it. So are those kind of new tools being used by, yourself, or in terms of evaluating, what's really changing? All of these products became very popular when we, started pulling back antibiotic use. And so that obviously shift the microbiome when we don't use antibiotics, and then we use these products trying to shift the microbiome in a beneficial way. So what's kind of the latest and greatest in terms of these high-tech tools, I guess, to evaluate what's going on in the gut?
Yeah, sure. And maybe I'll just back up a little bit and some background. As you mentioned, it's relatively new. It was not until the late 1990s where microbiome was really starting to be discussed. And I think more in humans than animals, and it's just evolved. So it was the late 1990s, which I don't think is that long ago.
Right.
But in the overall scheme of things, it is a very young science. And we learn more every day. There's so many bacteria, positive and negative, out there. And, not too long ago, it was just hard to analyze for all of them to determine what was there or not. So, it's molecular biology advancements, high throughput sequencing, the 16S, all of that is what's come into allowing us to look to see what is in the microbiome. And as we identify what's in the microbiome, now we can go look through literature and learn more about what each of those specific bacteria do, good or bad. So it is very new science still with that. And it took a lot of developments and knowledge in other areas besides poultry to get to where we are today with being able to even start looking at these. And you mentioned antibiotics, right? So we didn't exactly know what was going on when we were using antibiotics. And something to remember with antibiotics is they don't necessarily discriminate. They can kill good and bad bacteria.
Everybody in the pool.
Right. So we didn't have as rich or diverse or abundance in the microbiome when we used antibiotics versus what we have now. And I also like to say that as we've gotten away from antibiotics, every day is a new day. Every day is new territory because we've gotten another day, or however we look at it, another month, another year, further away from the use of antibiotics in a lot of our poultry. So we're learning more all the time. Something that we do is we use cloacal swabs and have those sequenced to determine what bacteria are within that poultry. And we would use we have a postbiotic product that we have available for the commercial industry to go along with this sampling or microbiota kind of analysis, however you want to think of it. So we would go in and do a sampling, swabbing, have all those swabs sequenced, determine what's there, good and bad. And then we have an artificial intelligence platform. We have a partner that has this artificial intelligence platform to, obviously identify these good and bad bacteria and determine where we should go if we see a lot of bad names kind of show up. And we can feed this postbiotic and then look at it over time periods to see, are we shifting that bacteria to be more positive, less chance of a dysbiosis situation. And with this product, it's a kind of a core component to it. And then we can customize it with different additives we have that kind of fit it together based on what we would find in an initial microbiome analysis, that we need to see more of certain good bacteria, less of some of the bad ones. And we can customize this product over time, and just kind of periodically, a couple times a year, doing a swab throughout a complex that would be on the product and make sure we're shifting the microbiome good, making it more rich, more diverse, and doing that over time is kind of what a tool we have. We talked about some, evaluations in research and in the field and field studies. This is one that we actually do. We've been doing in research, of course, to get our modes of action and gather data, build the database. But, it's definitely one we take to the field and want to take to the field to help producers and companies that produce broilers, turkeys.
Right. Yeah. I think that's the path that we're headed on is just the data is there. It's been a matter of that we don't really have the test, like evaluating that test, like the use of artificial intelligence, like that's, crazy to look at, what's in chicken poop, like that's really what it is we're trying to evaluate, what all those, good and bad bacteria. So in terms of like generating that data and learning what these modes of actions, I think that's the other piece that's always like kind of questioning for a lot of people is like, okay, I understand putting the good bacteria in there is good, but how do we like kind of focus down on, okay, this is actually doing this specific mode of action? I think that's a big question for a lot of people before they want to add these products in is like, okay, how is this working? Does it have any negative? Like, how do you get that, sort of information into the hands of decision makers?
Okay. Well, I have an excellent example of a research trial we did that we have been sharing. Of course, we try to publish and, share our data with potential customers as well. A recent research trial we did was in broilers, 0 to 21 days of age, and it was a coccidiosis challenge trial. So we grew the chicks for 14 days. We gave them a 10x dose of a cocci vaccine as their challenge, and then we let them go for 7 more days. And we did the microbiome analysis at the end of that 21 total days, or 7 days after the challenge. And of course, we had our postbiotic product, which is what we were interested in, making sure it's doing what it could do. And of course, the controls, we had a not challenge control, a challenge control. And then we also compared to a coccidiostat, popular coccidiostat on the market. And what we found was that, of course, we also did oocyst counts, right? Growth performance, oocyst counts. We found that the coccidiostat did do its job, or confirmed that, right, in reducing the oocyst counts. Unfortunately, but it's not the mode of action for these postbiotics. The postbiotic treatment group actually had the highest oocyst counts. But when we looked at growth performance, we had similar feed conversion ratio between the treatment group fed the coccidiostat and that fed the postbiotic. And those challenged controls did not have similar feed conversion. So when we go to look at the microbiome, we saw that one of the biomarkers that is associated with feed conversion ratio was more abundant in the group fed the postbiotic. So even though the mode of action of the postbiotic was not to reduce oocyst count, it does shift microbiome or allow bacterial biomarkers to show up that are related to performance and improving performance. So overall, we got the similar growth performance or feed conversion ratio, and as well as gains, but feed conversion ratio. So we had that specific example or specific biomarker show up there, and you could use that to explain the similarities we were able to get in feed conversion ratio. And that was just a 21-day study. So we don't know what would happen after that yet. But, we do have plans to carry that out at least into a, 42-day grow out or, more longer study. We just have not been able to do that yet.
Okay. And in terms of I guess just thinking my, physiology brain. So if your, Coccidia species can be fairly invasive. So if the two groups are, fairly similar and actually you saw an improved feed conversion with your postbiotic product, are the, sort of feeding the good bacteria helping to offer almost like a protective barrier from the Coccidia sort of invading, or any idea? I understand you're detecting a biomarker, but in terms of on a microscopic scale, are you looking at that in terms of okay, what's it really doing here? Are the bacteria binding the attachment sites for the Coccidia? Are we just shifting all of that so then the protozoa aren't able to attach? Any suggestion, I guess, maybe on how that's working on that level?
Sure. I don't have a definite answer, but just in some of my experience in the past and, some research literature, there could be something with some of these yeast products, prebiotics with protozoa and preventing protozoal problems. An example probably that's more documented would be Cryptosporidium in calves, showing, some of the prebiotic yeast components can help prevent attachment of the crypto. So in that, I guess I'm hoping to translate, though I have not shown that definite line, into some of poultry, like blackhead.
Right. Wouldn't that be great if we could stop blackhead? Yeah. We don't have a solution for that one yet.
Well, no, we don't. We're all working there. And actually, we've also been looking at turkey poult challenge studies with Histomonas with our yeast products as well as the postbiotic. And actually, we're going to repeat it, but we did see a reduction in transmission of the blackhead. So going to the cocci is where we started. I've seen some literature where Tenella we've seen some reduction in that by feeding these products. These prebiotics, are not digestible. They make it to the ceca. We know they're working in the ceca, which is where Tenella is, which is where salmonella is. So we kind of have that confirmed. So I think there's plenty of room for these in an overall program for cocci control. It's not probably the only thing we're going to be able to use, but I think they are part of a program for cocci control. That, like I said, there is some evidence with some protozoa that they, are effectiveness at preventing the harmfulness of those. So it's part of a program and something we definitely can explore more in the future.
Yeah, I think that I think that we do have to sort of figure out how to piece together a lot of these products. As as we keep getting things, removed from the market or for marketing reasons, things get taken away. It's we have to figure out what works, and what combination, yeah. And I think there's no one product that necessarily fits the bill and is like, here, just use this and it'll take care of everything. Right. So,
right. Well, yeah, we might have had that with antibiotics. And now we have all of these different feed additives that kind of go in, there's not one replacement. Sure. Another example I have is, we've kind of been looking at some combinations, prebiotics, probiotics, not just us, but there's a lot in the literature. We use a prebiotic, probiotic, and a saponin product, which saponins are effective against protozoa. The combination of those three together, we saw some positive biomarkers increase in abundance when we had the three together, as opposed to having just the probiotic, prebiotic together. When we added that saponin, we saw some advantages there as well. So again, a whole nother category of compound that we need to look at, and again, could fit into the big overall program.
Yeah, I think that's going to be our next steps is figuring out, how to, almost puzzle piece or, however, build these products to, address things like blackhead or, like, to me, Campylobacter is a prime example. Like, we really don't have great control measures for Campy. Is there a product out there? Maybe. Like, maybe it's, layering some of these things on top of each other. Eventually, like, we just don't really have great things for that. Like, everybody does a lot of research on salmonella, but Campylobacter's, just still sitting out there. We still have a performance standard. So, maybe there's an answer in some of these products.
Sure. I think there is. I think there's limited data, but there is some data in looking at the yeast components to reduce Campy as well. There's been some positive data there as well. But sure. Right. It's not necessarily going to hurt the poultry. So, where does it fit in?
We'll get more of that.
But yeah, we'll have to keep looking for the biomarkers that maybe can reduce the Campy or, those specific probiotic strains that can outcompete.
And I think another area where a lot of people focus on this is control of, sort of the clostridial types of infections in terms of necrotic and in terms of later on potentially dermatitis. So how does that fit in terms of, if we promote the good bacteria, how does that control clostridial populations? Because that's another sort of disease state that we try to minimize with these types of products.
Sure. Again, it's not, we don't have one tool that we can use to replace what had been used in the past. However, a lot of the bacillus strains are very inhibitory for Clostridium perfringens. They can prevent the growth, reduce the Clostridium perfringens. So I've seen a lot of the bacillus strains. When you feed that and you measure the Clostridium perfringens, you see a lot of reduction there. They can, again, be a part of the whole program and helping to reduce that necrotic.
So then I kind of have one last sort of area to sort of, cover in the conversation to pick your brain on, just so that folks understand at what time point can you impact the microbiome? Do you feed these products like at a certain age? Is it when the disease challenge is highest? Do you have to get in there at the very beginning? Sort of how do we make impacts on the microbiome?
Okay, so first we have to remember the microbiome is very variable. It's dynamic. Right? It's never, I don't think it's ever really constant, in a constant state. It changes. It can change daily based on what comes into the environment. It can change weekly. It changes with phase feeding, changes with each phase. So age affects it affects the microbiome. We tend to see different trends in bacteria increase or decrease as a bird ages. But, again, that can be to environment, feed changes. So it's something that changes all the time. So we need to be thinking we're using a lot of these biotics as preventative means of controlling something. So you definitely have to get them in from the beginning. There's products that can be used in the hatchery, whether you spray it on the chick itself, spray it on the egg shell, just to get a good bacteria kind of available there for that chick or poult, and help them get off to the best start. Because when they leave the hatchery, well, first of all, the hatchery, right, compared to if they had contact with an adult female, if they had contact with the parent, that parent would help them establish their microbiota. They don't have that. So unfortunately,
we hatch them in a building, not underneath the hen.
Exactly. And in those buildings, it's kind of like a hospital example. The worst survive no matter what we do. Even seen
the bad
hatcheries
bugs are super bad.
I've seen hatcheries go in and do a really deep clean and disinfection. The next hatch is just one of the worst, highest levels of E. coli. So we have to use these products as soon as possible. We get them through, get them hatched, get them to the farm. They come in contact with that used litter, hopefully has been treated to reduce the pathogen loads. But still, that litter is kind of its first probiotic, good or bad. So yeah, we need to get these into the bird, just, whether it's spraying on in hatchery, using as a water treatment at the farm right away. And because they're not going to, eat enough right there at the beginning either.
Their intake is so limited.
Yeah. Yeah. So, hatchery water can help get a good lactobacillus establishment there in the gut. And then, like I said, they're preventative measures for the most part. So the entire life cycle is when they're beneficial, to help, you have times when they're under stress. So you have shedding of these pathogens. So just kind of, and when if we know they're undergoing a stress, or, maybe it's just a diet change, which can be stressful to them. Maybe we increase or provide extra probiotic in the water, another water treatment, or, we know a history of when a certain farm or group of farms breaks with necrotic. Okay. Prior to that, we increase the probiotic, or again, can go in the water system. So, there's all kinds of options. But again, the entire life cycle of that bird, these have a role and can play, be a benefit. Maybe it's preventative. We would never know if we didn't have that in there. Maybe the birds could perform the same. But if they come under that challenge, you have that protection there within the bird to hopefully get them through it, overcome it, and maybe the producer won't even know there was a problem. That would be the ideal world.
That would be great if we could get it that fine-tuned to, almost a personalized treatment program per farm, because it's so specific. I think that was a perfect spot to wrap up our conversation focused on the microbiome today.
Your partner in improving animal performance, Berg & Schmidt. They believe the following additives are necessary in the poultry dietary: functional lipids for an efficient dietary energy management, phospholipids for emulsification achieving a better nutrient intake, MCTs to provide energy and modulate the microflora within the intestines and enzymes for elevated use of fibrous materials and byproducts.
So I'll ask just kind of an off question. What advice would you give people, that are just starting on a similar path in the nutrition world, or, supporting products like what you support?
One of the things we learn to do as students is we learn how to learn. So never stop learning. Education, I was an educator. I guess I still consider myself an educator.
You still are. Tech service is a lot of educating.
Yeah. Yeah. So, take advantage of that education, never stop learning, and, make as many connections as possible. Obviously, with the faculty, a graduate student learns builds relationship with their major professor and the other faculty within a department that they're working in, as well as other graduate students. We learn from each other. So, and then take advantage of scientific meetings. Obviously, that's where there's a lot of knowledge shared. You'll see all kinds of data shared. So, kind of look at it, learn from it, and take those opportunities, when you're at a scientific meeting to meet people that, you're reading their research, and try and have conversations with them, and, just make a connection there. It's the poultry industry is a big industry, but it's also small as well.
Very small world.
Tend to know each other, right? So, I just say, just take advantage of all the educational opportunities you can, and never stop learning, because there's changing. We've been talking about a newer type science here today that we know some about, but I'm sure there's a lot we don't know yet.
Changing I feel like every week I learn something new in this whole arena of gut health and the microbiome. Like, even though I consider myself like a diagnostician, like I do disease, but like that's a big part of it. So it's, I lifelong learning, great way to end. And we appreciate your time today, Thresia, and thanks for being our guest today on the Poultry Podcast Show.